Can you see the presentation now?
Yeah, it has started. Right.
Sure. Perfect. All right. Thank you so much, Dr. Pathak, for a kind introduction. It is my pleasure to be in company of you all. I would also like to thank the Biochemistry Department and Ram Manohar Lohia University for giving me this opportunity to present in this forum in this such a relevant topic of COVID fallout, COVID-19 fallout. COVID-19 has impacted or has had a very significant impact on all aspects of human life, be it biological, physical, or spiritual. Some of the impacts have been positive, most of, as we are familiar with, have been negative. And one of the positive impacts of COVID-19 has been the new and creative ways of doing research, conducting business. And an example of that is our webinar today. We are having a virtual international seminar or webinar, which has been carried out by our university virtually, very conveniently from our home. Another positive impact of COVID-19, there are not many positive impacts, but one which has really stood out has been the general awareness and general curiosity of general population to understand the disease, to understand the development of the therapeutic, to be involved basically in the research community and the progress of the research for therapeutic as well as so much so that vaccine development and tracking the development of vaccine has become a topic of general conversation, household conversation. So with this background and observing this impact, I decided that for today's presentation, I will focus on vaccine development and review of vaccine through COVID lens. Okay? So the presentation will. So the presentation will mainly focus on the vaccine development for COVID-19. But I would like to start the presentation by giving a general overview of vaccine and vaccine development process, contrasting it with how it has evolved during, or how the changes have been implemented during the vaccine development for COVID-19. So what are vaccines? As you know, the crowd here is probably very familiar with, vaccines are preventive medicine. They are basically either inactivated or part of a pathogen which stimulate the immune system in much the same way as an actual infection, with the goal being to mimic the infection without the side effects or the serious symptoms of the disease. A successful vaccine activates T and B cells through adaptive immunity. A vaccine is taken up by antigen-presenting cells. It activates the T helper cells. T helper cells in turn activate the T and B cells. The T cells mount an cell-mediated response, killing the host cells which are harboring the virus, and also stimulating other immune cells of the body to destroy the virus. The B cells, on the other hand, produce antibody molecules which are specific to pathogen. These antibody molecules bind to the pathogen and prevent the entry of the pathogen into the host cell, as well as marking this pathogen for destruction by other immune cells. So these activated T and B cells, they are not a very long-term immune response. They mount a short-term immune response. A fraction of these activated T and B cells also become memory cells, and it is these memory B and T cells which are responsible for long-term protection against the pathogen. On a subsequent introduction of the pathogen in the body, these memory T and B cells can very quickly mount the antibody-mediated or cell-mediated response and thereby prevent the progression of the disease. That is just a generic overview of the vaccines, how they work. So besides protecting the individual or general population against the disease, during a pandemic or during the infectious disease spread, vaccines also have a very important role of helping control the spread of the disease. And that is carried out by reducing the number of individuals that can further go on to spread the disease in the susceptible population. A phenomena which is referred to as herd immunity, which has also become a very common term. So vaccines control the infection or spread of the infection by mediating herd immunity. So vaccine development is a very, very long and an expensive development process. The traditional vaccine development is a very long, very expensive process. A typical vaccine development cycle lasts anywhere from 10 to 20 years. And so I see that somebody has raised a hand. Is it okay if I answer the question at the end so that the flow of the talk is not interrupted? Yeah, so if you can hold on to your question, I will get to your question at the end of the presentation. Thank you so much. All right. So the vaccine development is a very long and expensive development process. It lasts, typical traditional vaccine development process lasts anywhere from 10 to 20 years. Also during this development process, there is a huge attrition of the candidates that are being pursued. So it is a very common practice for vaccine developers to have multiple candidates, to be working on multiple candidates simultaneously, and also to go through this development process in a sequential manner. That is, they move on to the next phase of the development after successful completion of the previous phase of development, with having ample time in between the two phases so that they can review the success of the previous phase and have adequate checks and balances before they go into the next phase of which again leads to a long development cycle. A typical development of vaccine is captured very well by this cartoon, if you will, from FDA's website. A typical traditional vaccine development will start with animal model studies. Some kind of preclinical studies which are carried out in an animal model system which can mimic the disease created by disease as manifested in the humans by the pathogen. After the successful completion of the preclinical studies, the vaccine drug then moves into the clinical phases of development. During the first phase 1 of the clinical stage of development, typically the dose escalation studies are carried out with limited number of volunteers. Basically, multiple doses are tried out, and the safety is evaluated with different dosages. After the phase 1 study, a dose is set, and then with that set dose, phase 2 studies embarked upon, during which the set dose, the individuals, a much bigger cohort of volunteers is immunized with the set dose based on the previous phase of the trial, and again safety is assessed at that particular, more extensive safety is assessed at that phase of the development, at that dose. After completion of the phase 2, then the vaccine moves to the phase 3 stage, which is a more controlled, placebo-controlled study, and a much larger study where the safety, detailed safety as well as efficacy of the vaccine is evaluated. So it is a multiple phase study, multiple phase development involving a lot of enrollment of volunteers. After the phase 3, the vaccine then goes through the approval process through the regulatory agencies, which is also a lengthy process, has a lot of checks and balances for assessing the safety, evaluating the safety of the vaccine before it is released or for general administration. The development process, this long development process cannot support the development of vaccine during a pandemic. So during a pandemic, the process is compressed significantly. Various steps of the development cycle are compressed. The vaccine, usually the developers start off with a fast start, either completely eliminating the preclinical studies or carrying out shortened preclinical studies, and also executing all the different phases of development in parallel, without completion of the previous stage, or without assessing the successful outcome of the previous stage. This whole compression is also included in the manufacturing process. The manufacturing is scaled up to commercial level of development even before the clinical safety or clinical outcome of the vaccine is assessed. So during a pandemic, the development cycle or development time frame is significantly reduced by compressing the various stages of the development process. So far, based on the vaccines which are in development, based on monitoring their progress, some of the deviations from the traditional vaccine development that various vaccine developers have implemented in their process, as there are no known when the virus the infection started and the disease started, there were no known animal models for this disease. So not all drug development, not all vaccine developers are carrying out the preclinical studies in animal models. So far, the various drugs that have reached phase 3, they have had very small phase 1 and phase 2 studies, and very often phase 1 and phase 2 studies have been combined. And the vaccines which have moved into the phase 3 are also relatively small, with tens of thousands of people only enrolled for the phase 3 study. The approval process by the approval agencies and the various regulatory agencies has also been modified significantly. There have been significant reduction in the administrative process that have been associated with vaccine development, with limiting the a lot of advisory committee meetings and so on and so forth, and also empowering emergency use authorization of the vaccines to the regulatory agencies. Similarly, manufacturing, as you all are aware of, most of the vaccine developers, as they are going through the process of developing, are also heavily involved in securing manufacturing facilities, manufacturing collaborators. Almost all the leading vaccine manufacturers have some collaboration with big manufacturing groups for the scaling of the manufacturing, even before the vaccine has been clinically has completed the clinical test. So now moving on to the virus or the pathogen, which is at the center of this disease. So that the virus which causes COVID-19 belongs to the coronavirus family. There are 4 commonly circulating members of this family, which can infect humans. They cause these 4 common circulating strains or members of this family cause very mild upper respiratory tract illness. They, as I mentioned, they are commonly circulating. The human body, most of us have at some point or another have got infected with one of these viruses. There are some variants which are basically the coronavirus viruses which infect animals and can sometimes jump across species and have the ability to infect humans. And these variants, when they cross the species, they actually result in more severe form of disease. And SARS-CoV-2, which is the causative agent for the COVID-19, belongs to that variant strain of the coronavirus. So SARS-CoV-2 is a single-strand. It's an enveloped virus having a positive sense single-stranded mRNA as its genome. Its genome size is 25 to 32 kilobases. It has four major four structural protein. The spike protein, which gives it the characteristic image of projected spikes or corona-like image. This spike protein is a glycoprotein that is expressed on the surface of the virus. It is this protein that mediates the binding of the virus with the receptors on the host cell. Within the protein itself, specifically, it is the receptor binding domain that mediates the binding of the virus to the host cells through ACE receptors. So because of this role of spike protein and RBD, receptor binding domain, the protein as well as the domain are very attractive vaccine agents and are being pursued. These are the two major forms that are being pursued by most of the vaccines. So this is a current status of the various vaccines for coronavirus or SARS-CoV-2. There are currently about 170 vaccines in development worldwide. This image is from a New York Times vaccine tracker, New York Times website, vaccine tracker that they have on their website. But I actually cross-validated this with the WHO website as well, the WHO resource as well. So these out of these 172 vaccines, 6 are in phase 3 clinical trial. 1 has got emergency use authorization, and I'll talk about that in a bit. And the rest of them are in various stages of. So these 172 vaccines, they are, or the developers of these 172 vaccines follow one of the 7 listed or 6 listed approaches for developing, for producing the vaccine. And out of these approaches, the first 3 have been successfully used or are used in the vaccines which are currently approved, are currently in use for various diseases. And the last 3 approaches are new and more investigational and are more actively being pursued for the first time for SARS-CoV-2 or COVID-19. So in the next few slides, I will go through the various vaccines as they relate to these approaches and talk about their current status, where they stand as of now in the development. So let's go to the next slide. So the first approach, which is quite a common approach, is the whole killed virus. In this approach relies on using killed virus, killed pathogen, or inactivated pathogen as an antigen for eliciting immune response or for stimulating the immune system. This is as I mentioned, this approach has already been successfully used for polio, hepatitis A, and rabies vaccine. One of the advantage of this approach is that since it is already being used for these vaccines, there is already existing knowledge base for regulatory agencies or also for the development, for manufacturing and for development, so which can potentially accelerate the development of vaccine following this approach. The one big limitation of this approach is that during the manufacturing stages, large amount of infectious virus needs to be handled, which can require special handling and can be expensive. This approach is being followed by Sinovac in China and Bharat Biotech for their lead candidates. The Sinovac candidate has successfully completed phase 1 and phase 2 study and is in phase 3 right now. Bharat Biotech's candidate is in phase 2. And there is no published work as such, at least in the peer-reviewed journal from the Purified candidate. The Sinovac has published the preclinical data from, has recently, there's a recent publication from animal studies, they published the data. But the phase 1-2 data from their vaccine lead candidate was shared as a press release. And this is also this sharing of information, sharing of scientific information or the status update through press release is also one of the key deviation during COVID-19 research in research phase. There is lack of, people are publishing or sharing work without going through the whole peer review process and in scientific journals. So that has been one of the pain point or sore point for people who are following vaccine development. So in any case, the Sinovac shared their successful outcome of, or success of their phase 1 and 2 data from their phase 1, phase 2 study with 743 participants. They through a press release, and they said that there was no significant adverse effect observed in this study, besides some mild manageable symptoms like fever, chills, and pain. They also mentioned that the antibody, almost all the participants had neutralizing antibody response. The details of the antibody response titer-wise were not shared. In the publication that recently came out in journal Science, they shared detailed data from their animal studies, and in this publication they showed data from mouse, rat, and non-human primate vaccination, neutralizing antibody response, as well as protection against the disease in non-human primates with using two different dosages of the vaccine, then challenged with the SARS-CoV virus. So this was a good study, preclinical study information. So the second approach, which is again has been used successfully in the past with vaccines which are already currently in use, is live attenuated virus. This approach uses techniques to uses the live virus combined with techniques to attenuate the virus or subside the infectivity of the virus. One of the advantage of this approach is that it is one of the most effective approach in vaccine development. So it elicits one of the strongest and most robust immune response, and as I mentioned, has been used in many successful vaccines. The limitation of this approach is that because the attenuation virus needs to be attenuated, various different approaches have to be tested out for that attenuation, for reaching the attenuation of the virus, and also extensive safety analysis needs to be carried out. So this approach can be one of the longest approach for developing a vaccine. And Codagenics in New York, in collaboration with Serum Institute of India, is pursuing this approach for their candidate. And since this is a very has a very lengthy development cycle, the approach is still in preclinical stages of development. All right. So the S protein, which is, as I have already mentioned, S protein and the receptor binding domain of the S protein are a very attractive target for vaccines. So they have been they are being currently investigated as purified viral protein vaccine. Again, this approach also has been previously successfully used for hepatitis B, HPV, and influenza vaccine. This approach involves production or generation of the antigen, the protein, recombinantly in vitro. So the limitation could be, one of the biggest limitation could be handling the production capacity, and as well as demonstrating the integrity of the antigen, which has gone through the recombinant development process. This approach is being pursued by Novavax and Sanofi Pasteur. And just recently, earlier this week, Novavax, again through a press release, announced positive phase 1 data from, I think it was 131 healthy adult studies using 2 different dosage of the vaccine. They also shared that the vaccine induced a neutralization in almost 100% of the participants without having any significant adverse effects. There were a few hospitalizations, but they were not the details about those hospitalizations were not shared. There is no peer-reviewed published update from either of these groups that I can share at this point. So now moving on to the approaches which are novel or not, for which there are no examples of previous vaccine. Viral vector is the viral vector vaccine is one such approach. There is, as I mentioned, there is no previous approved vaccine under this category, but this is it is one of the. There are some investigational investigational studies for emerging viral diseases following this approach. For example, for Ebola and dengue. And in those investigational studies, this approach has shown very strong effectiveness and very strong immune response. Under this approach, unrelated, non-pathogenic virus is used as a vehicle for delivering the antigen coding sequence from the pathogenic virus. Typically adenovirus or similar non-pathogenic virus or other non-pathogenic viruses are used as this. This approach is being pursued by University of Oxford AstraZeneca and CanSino Biologics. They have both their candidates are in phase 3 stages. In CanSino actually published data from their phase 1, phase 2 study. But before the vaccine even progressing to the phase 3 or sharing data from phase 3, Chinese government provided emergency use authorization of this vaccine for administering to military. So this is the only vaccine so far which has got approval for administering to not general population but on priority basis. But both the leading vaccine developers in this category have published their work in peer-reviewed journal. So the first, actually, CanSino should be credited for providing first peer-reviewed data for their candidates. So this was the first publication in peer-reviewed journal for any vaccine. It was published on May 22nd in the journal Lancet. They shared, this was a first in human trial, phase 1 trial. The CanSino is using adenovirus 5 vectored COVID-19 vaccine. They show in this paper, they showed that the vaccine was well tolerated and was immunogenic in healthy volunteers. 1 dose of the vaccine at all concentration led to immunogenicity, and it also elicited a T-cell response as well. There were some mild symptoms like fever, muscle pain, joint pain, which were manageable by using common drugs which are used for treating symptoms. All right. So the second developer following this approach of the University of Oxford, in collaboration with the AstraZeneca, they are using the chimpanzee adenovirus as the vehicle and their vaccine is based on chimpanzee adenovirus. They also have published their work in from the phase 1, phase 2 trial in the peer-reviewed journal Lancet. And they also shared data about safety and tolerability and immunogenicity of this vaccine in a single dose with both humoral and cellular response. both CanSino and University of Oxford vaccine are now progressing in phase 3 clinical trials. So, and the next approach in this novel approaches is the DNA vaccine, basically delivering the antigenic DNA, the DNA that is coding for the antigen directly to the human cells. And this approach is relatively very novel. And if there is no previous licensed vaccine and very limited data is available, and DNA or nucleic acid in general, it's very difficult to introduce into human cells. So it requires the one limitation of this vaccine is requiring specialized delivery like electroporation and specialized devices for delivery of this vaccine. The other shortcoming of this approach is that because the DNA has to be taken up by the cell and then the nucleus and then have to get transcribed and translated, typically the vaccines, so far the vaccines which have been tested, have shown low immunogenicity. Inovio in US, Osaka in Japan, and Cadila in India are following this approach for their lead candidates, and all their candidates are in phase 1 and phase 2. There is no published data as yet from them for sharing the status. And moving on to the last approach, which is a very hyped approach, at least in US, because of the lead player in this field being Moderna company, which is in US, which has blessing of the NIH as well, is the RNA vaccine. In this approach, the RNA molecule, which is going to translate the desired antigen from the pathogen, is directly delivered using the various delivery approaches into the human cell, and then goes through, is taken up by either the antigen presenting cell directly or other cells of the host, and goes through translation and produces the antigen for mounting the immune response. One biggest advantage of one biggest limitation of this approach is that, as you all are familiar, RNA is a very, very labile molecule. So managing its stability during manufacturing as well as development is a big challenge, as well as managing the delivery mechanism, through using nanoparticles or lipid encapsulation. There is not much experience available as yet for implementing those manufacturing processes as well. As I mentioned, Moderna in US and Pfizer and BioNTech from Germany are pursuing this approach, and they are quite advanced actually. Both their candidates are in phase 3 stages of development. Both have also published data from their phase 1 and phase 2 study in peer-reviewed journal. This publication is from Moderna's phase 1, phase 2 trial, which was a relatively small trial with only 45 volunteers. The study was published in New England Journal of Medicine. The mRNA vaccine encodes for S protein of and the SARS-CoV-2, and in this 45 and in this study, in this phase 1, phase 2 study, they used 3 different dosage of the vaccine and elicited neutralizing antibody response in all participants, and there were no significant adverse effects besides the usual fever and malaise associated with this vaccine as well, which warranted the progress of this vaccine phase 3. The Pfizer and BioNTech actually have probably one of the till date the most robust response has been reported by the candidate being pursued by Pfizer and BioNTech. Their phase 1, they have conducted 3 different phase 1 and 2 studies at 3 different locations. So it's a very exhaustive phase 1, phase 2, more exhaustive, not very exhaustive, but more exhaustive than other approaches that they have done phase 1, phase 2 evaluation. The vaccine, their vaccine candidate codes for the receptor binding domain, And in this study, which was published in a preprint, actually, this was not in a peer-reviewed journal, Medrex, they shared data for their candidate PNT6162B1. They tested several different doses. They showed that the vaccine was well tolerated and generated dose-dependent immunogenicity, which was measured by ELISAs as well as microneutralization. And the titers of neutralizing antibodies were quite robust for the BioNTech vaccine. So in summary, I would like to conclude the presentation by saying that research community, the scientific community, scientific research community has really risen to the challenge of developing a vaccine in a very short time frame for a very serious disease indication. And I'm very hopeful, and so far based on the data shared and the data published so far, I'm very hopeful that we will have a vaccine approved or released very soon in few months, early next year or middle of next year. However, there are some key considerations that should be kept in mind before we pin our hopes too high. This is the most of the phase 1, 2, 3 trials are really short-term trials for these studies, and have with very few, much reduced number of volunteers. So there is, even after the approval or release of the vaccine, rare adverse effects, adverse events can happen, and it has happened before with the, I believe, measles vaccine. It has happened before, and we can expect that in this case for sure because of the reduced development time. The other consideration to keep in mind is the kind of immunity that will be developed, because again, the development cycle is short. The immunized or the volunteers in these developmental phases are not followed for long term, and there is lack of information about the length of the immunity or that is imparted by the vaccine. So we don't know how long whether the immune response elicited will be long term or relatively short. And again, another thing that should be kept in mind is that this is a very, very novel virus, and the disease is also very new. We are still developing our understanding of this disease. We are still developing our understanding of how the immune system responds to SARS-CoV-2. So, antibody mediated enhancement of disease can also happen, or immune response can also happen. So how the immune system will respond to the vaccine as well needs to be evaluated. The efficacy, whether the vaccine is going to be completely efficacious or partial efficacious, that needs to be evaluated. And also because most of the approaches that I shared are relatively novel approaches, manufacturing related challenges, which actually Moderna is already struggling with, can also hamper the development or affect the timeline of the vaccine. So with that sober note, I will end my presentation, and I'll be happy to thank you all for your time, and we'll be happy to take it.
Thank you, Dr. Gunjan. A very wonderful talk and making us aware about various strategies of development of vaccines for COVID-19 and a latest status also. Hope soon vaccine will be developed and available to all, and this trauma will be over. And now the forum is open for the question. Participant, if you have any question, you can ask.
Well, very good morning to Dr. Gunjan Tiwari. I joined the session late. Am I audible?
Yes, I can hear you. Good morning, Dr. Jamal.
Thank you very much. Very good morning. And it was a wonderful deliberation regarding the vaccine program, which is in the offing and which is progressing a lot. To me, it seems that RNA vaccine is may not work too much as compared to the DNA vaccine. Is it true?
It could be, again, both DNA and RNA vaccines are new approaches, and we don't have previous experiences from both. The advantage of RNA-based vaccine is the delivery methods that are being used. The encapsulation, lipid encapsulation, nanoparticle encapsulation. At least they are demonstrating so far, at least in phase 1, phase 2, they have demonstrated effective delivery. We have to yet see how the DNA vaccine performs. I haven't seen any data as yet. The only challenge I see with the DNA vaccine is the delivery. What would be the method of delivery? And again, based on my understanding of science, what I feel is DNA has to cross the plasma membrane, the cell membrane, and then the nuclear membrane to get into the nucleus, get transcribed and translated. So that, thinking of that, keeping that in mind, I feel that DNA vaccine might have low immunogenicity as compared to mRNA or RNA-based vaccine, which can be translated right away once they get into the cell.
Madam, actually, you see, in case of the HIV virus, there also the RNA is playing a very crucial role in the multiplication and proliferation of HIV particles. Here also the situation is the same. And as far as the theoretical part of is concerned, like replication, transcription, translation, the rate of mutation, the chances frequency of mutation in case of the RNA molecule is much higher as compared to DNA. So stability, as far as if we compare the stability of the molecules, they'll find that DNA molecule is much more stable as compared to RNA molecule. And therefore, you will have a cocktail of vaccine, a cocktail of molecule for the vaccination program, because one molecule is not going to work. See, if the epitopes undergo change, then you need to have a cocktail of things to work. Because some epitopes are strong epitopes, weak epitopes, and some are masked epitopes, and all those things, all those phenomena are there. So keeping this in mind, I think the research area as far as RNA vaccine is concerned, or if we use any virus which is based on RNA replication, RNA which is having an RNA molecule for multiplication, then any vaccine is not going to last for too long. So research in this area will continue for a very long period of time.
Absolutely. You are absolutely right. If nothing else, at least this whole process will shed a lot of light into all these new strategies because of the rapid development and because of the amount of funding that is being poured in. At the end of this, COVID-19 vaccine exploration efforts, at least we will have a more clear, we will have a better, and that's what I'm hopeful about, that we'll have a better understanding of how these different approaches work. As of now, so my presentation was based off of, at least for the COVID-19, it was based off of published work, what I could find what I could put my hands on.
That is true. It was an excellent presentation, no doubt about it. But COVID-19 pandemic has come to teach us a lot of things.
Absolutely.
Okay. So by the time we are in the stage of having a successful vaccine, this coronavirus may disappear altogether.
Absolutely. As I said, our immune system we are still understanding how the immune system responds to this virus. So you are absolutely right. For all you know, we would have understood the disease much better, and we would have come up with disease management options rather than resorting to the vaccine.
Thank you very much. Over to Dr. Pathak, please.
Participants, do you have more questions you can ask? This is a very interesting lecture and very relevant.
Okay, we can move on to the next talk.
Yeah.
Thank you so much. Yeah.
Thank you, Dr. Gunjan.
It was a pleasure. Yeah, it was a pleasure talking to you all.
Yeah, thank you for sparing your valuable time and joining us. Okay, thank you. Thanks a lot. Thank you. I think our
Professor Rajat Sandhir, sir. Am I audible to you?
He's here. I don't think he's here.
Rajat Sandhir, sir, are you there? Not yet. Actually,
Not yet.
His talk was scheduled from
10. And it is 8 minutes to go.
Let me give a call to him. No, he has told that he will be joining after at 10 o'clock. Professor Rajat Sandhir will be joining at 10 o'clock. So have patience till then.
Yeah, it's already 9:52.
Yeah, he said he
8
requires
minutes to
another 7 to 8 minutes more.
Okay.
So, Ramesh, Engineer Ramesh Mishra.
Yeah.
You can put the things on mute till 10 o'clock.
Yeah.
Professor Rajat Sandhir, can you hear me? Okay, he is logging in. Just a minute. Rajat sir, can you hear me? Am I audible? Hello, good morning.
Good morning, Farooq.
Good morning, good morning.
Finally, I'm back to you there in virtual way.
We welcome you with open heart.
Thank you, thank you, thank you.
So over to Dr. Neelam Pathak. Professor Neelam Pathak, please take up the talk.
Good morning, sir.
Good morning, good morning. How are you?
I am fine, sir. How are you?
Good, 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 good.
Welcome you, sir, in Dr. Ramanohar Lohia, Awadh University, Ayodhya. Professor Sandhir, he's a professor at Department of Biochemistry, Punjab University, Chandigarh. He has received his MSc and PhD degree in biochemistry from the Postgraduate Institute of Medical Education and Research, Chandigarh. He has been with the Punjab University for more than 20 years. His research interests are to understand the biochemical and molecular mechanism involved in development of neurodegenerative conditions like metabolic encephalitis, dementias, brain injury, with particular interest to understand role of oxidative stress, mitochondria, and alteration in blood-brain barrier. In addition to his interest is also to identify neuroprotective strategies that could solve neurodegenerative conditions. And he has over more than 175 papers to his credit and has mentored over 24 students for PhD. He has an H index of 39 and I-10 index of 92. He has been elected as Fellow of Indian Academy of Neurosciences, Fellow of Indian Association of Biomedical Scientists. He has been served as editor, Punjab University Research Journal, member editorial board of World Journal of Diabetes and Toxicology International. He was also nominated as executive member of the Indian Academy of Neurosciences. He has also served as a chairperson department of biochemistry, also coordinator of Institute of Forensic Science and Criminology, coordinator of Center for Nuclear Medicine, and he also served as head and coordinator Central Animal House, and fellow of Punjab University Senate, member of Research Promotion Cell, and he is a member of various scientific society, few of them, Indian Academy of Neurosciences, fellow of Indian Association of Biomedical Sciences, and he has been also nominated as executive member of the Indian Academy of Neurosciences. With this, I very humbly request Professor Sandhir to deliver his talk on innovation during COVID and pandemic. Over to you, sir.
Wait a minute, ma'am. Brother Sandhir sir, I would like to add two more things. Firstly, he is a very good auditor, and secondly, he has been very instrumental and a very key person for the Department of Biochemistry at Dr. Ramanand Lohia, our University, Faizabad. So over to you, sir.
So good morning to you all. I would say coming on the last day is always a challenge. And when you have this COVID as the topic, so I would not like to remember anything that the biodata said. I only know that I am connected to our university, Faizabad, where I got nurtured. And I'm going to say a honest statement here. The students from this university have been the best ever in my career. And I said this to the previous vice chancellor also. And I've seen over my 25 years of journey that you have the best potential in this part of the country, I would say. And I would say the Department of Biochemistry, our university, I'll say it in brief, but I know we have a longer name now. I would say it made progress in 5 years. I came back to a 50-year-old department, and I have no hesitation in saying that Faizabad department made more progress in 5 years. So I would say team effort is the only key that we did. And I was a young boy when I started my career there. And I would say when we would conduct the practicals there, sometimes the vice chancellor would walk in and say, "Where is your teacher?" and that would be, you can understand what so that is why I said we were nurtured there. And you probably got the unfortunate news yesterday. We lost a colleague. Out of 7 of us, we are now 6 of us, and we started our careers almost the same time. So it's a sad note, as well as I'm happy that I'm again connected back to my roots. Actually, I would say these are my roots, because there's a lot of journey that we traveled together, and there are positive experiences from Faizabad that transformed my career, I would say that. And we started from one small room, and I've seen the department, I came back to the department maybe 3-4 years back, or 2-3 years back, and I found the university campus is even transformed. So I'll not say too much now, because some of you may be waiting for what I have to discuss. I would say you had an excellent scientific deliberations in last 2 days, and I was following the kind of feedback you gave. That might make me nervous. So I'm not an expert in the field, but I've just tried to bring in my thoughts on the topic, because everybody is shifting their focus towards what is required and relevant to the research now than what I was doing a couple of years back. Even I am thinking of changing my career or shifting my research interest towards COVID in 2 ways. I am even thinking of starting a company which can spin off and give a product that would be there in the market. So we all have to think of something that is useful for society and humanity at large. So with this, I would just like to get started with my formal presentation, and I would like to thank Farooq and Dr. Neelam Pathak for taking the flag of the department forward. I would say this is our department. I don't remember that I'm in Biochemistry Department, Punjab University. So I will still own this department forever. I would say that. Thank you. Thank you.
Thank you, sir.
So I picked up this topic, innovations during COVID-19 pandemic, silver lining in a black cloud. We've all gone through this journey in last couple of years, and we know how this has changed our lives immensely. And I will not even spend a minute on this slide because this slide is very familiar to all of you. This is the beautifully looking virus, which is almost brought the life to standstill. And you know, there are spike proteins, there are membrane proteins, envelope protein, nucleocapsid proteins, and lipid membrane. And again, bit of history of the virus. And you probably, I will not get into there. I'll just emphasize on one fact. There have been viruses that came earlier also, severe acute respiratory syndrome, Middle East respiratory syndrome. These are the viruses that came in the recent past. And in last 6 months, this virus emerged in December from Hubei province in China. And you see it is impacting all spheres of human life. And this is the current global spread of this virus. This is from yesterday. You can see almost all parts of the world have been touched by this virus. And we have now over 200,000 cases worldwide. And look at the number of deaths. We have over 700,000 deaths worldwide reported from this. This is the same data presented in numbers, but look at the bottom part. It tells you how India is, the numbers in India are escalating. We have over 20 lakh cases in India, 41,000 deaths, and there are active cases which are over 6 lakhs. And look at this statement from India Today, the Times of India yesterday, saying India's COVID tally in August is highest in the world so far. So we need to worry. There is a challenge that this virus has thrown to the humanity, and we all would emerge victorious, I would say. But at the moment, we don't see any end in sight. And if you follow the graphs, I want to emphasize on two things in this graph. Look at South Korea. They had the ones who got this virus early on, and see how the graph is flattening in their case. Yesterday also, they had 14,000 cases, whereas look at the graph, which is the red line is for India. The numbers are increasing, and we have almost 60,000 cases per day reported from India. This is the projections that have been given. They say by next February 2021, we probably would be having over 287,000 cases per day, and which will be far more than any of the countries that are still infected, having virus infections. So being a larger country, it's a challenge for us and for humanity at large, I would say. We are all familiar with this picture also. It almost stopped our lives. It stopped the world for some time. And now we are recovering, although we know the virus is not gone, the danger is not gone. But how long we can go into lockdown, that's something that, there are reasons that these kind of decisions are taken, economic decisions and considerations, and there are other considerations that would come in. You don't want people to die of poverty if you are controlling the virus, because people will be jobless, all the industry would come to standstill. These are the compulsions that led us to open up the country, and that's one of the reasons that the virus spread is even more. Look at some kind of interesting stats. 120 nanometer strand of RNA, which weighs almost 0.85 attograms, 10 raised to power minus 18 grams, has brought humankind to knees. 70 billion viruses are required to make one person sick, and the weight is, look at, in decimals. And 2 grams have been estimated to infect 4 million humans. And you can say the world has been turned upside down. And we probably feel the world may not be same ever again. And we say there are new normals that we have to accept them now. And one of the new normal is we are also meeting each other through virtual mode. So how has COVID changed the world? It has impacted healthcare, economy, education, tourism, environment, agriculture, entertainment. Everything is impacted by this virus. And so I would say it's been a shock. to society, healthcare system, economies, and governments worldwide. And we need to come up with ways we can manage this crisis. And you know how miserably World Health Organization has failed. They've been coming up with updates almost every fortnight, giving a new input on the way we have to handle the virus. But to handle this crisis, we have to come up with the way that we can be back to having normal economy, we can have sustainable world, we can have our population who can work normally. And this is the time when there is a crisis. We all know when Indians are better at that. During crisis, we innovate, we are more creative. I think if you push a person to the wall, we know that the person rebounds, or the humanity even rebounds. And all efforts of the world are now to come up and create, innovate, so that we can fight with this challenge together. So all for the students, I'm saying, what is innovation? Innovation is the process of translating an idea or invention into service that create value. Value could be saving lives. It could also be generating revenue. And the pandemic is turning out to be a universal wake-up call for all such innovations. And I'm going to share some of the innovations that have come up recently, and I will touch upon different fields. And my idea is to sensitize you, not to impart you with too much knowledge. You had been bombarded with knowledge in the last 2 days, I guess, and I would just try to sensitize you. So first and foremost thing for innovation is creative thinking, and that involves novel and original ideas or solutions to solve problems. And what Albert Einstein said some time back is imagination is more important than knowledge. So what do you imagine? And I always say in science, you need imagination like that of a poet. And I think these are quotes, patience like that of a mule. And there are a few other attributes. I would say patience and imagination are two key things that you need for good research or good innovation. For knowledge is limited to all, we all know and understand, while imagination embraces the entire universe, and all thereover will we be to know and understand. This is Albert Einstein said, imagination is more important. If you imagine something, you probably can convert that imagination into something real. And creativity needs novelty. It needs something that is non-obvious, that means non-apparent, and appropriateness. It should be something which is constructive and socially desirable. And there are barriers in creativity. I would say some people, we also have to break our habits sometimes to be creative thought thinkers. And we know that in our school system, at times we try to suppress creativity by asking people to go for rote model rather than coming with innovative ways of things. And then you need motivation, emotional support for critical thinking. There are cultural barriers also for creative thinking, and perceptual barriers also to this. But the blocks can be overcome. If you know there's no other solution, you will find a way out of this. And that's what is driving innovation now during times of COVID crisis. An interesting quote I would like to share with you. Khush raho lekin kabhi santusht mat raho. For researchers, I would say never be satisfied. I think this is true for in other spheres of life also. If you're satisfied, probably you will not push the agenda forward. So I would say that desire to have more is something that would drive the agenda to be innovative. Being from neuroscience, I would like to say there are different faculties that we have in our brain. And if you see, the left brain is more towards logic and the right is towards creativity. And imagination, emotional intelligence, expression, everything is coming in there, and that's probably helping. So that means it is the right brain that would push our agenda towards innovation when we talk of innovation. And to be creative, we need to be alert so that we are aware of what is happening around us. habit of wider reading. If you read from different disciplines, probably you will connect disciplines, you will connect things, and probably, and the most important, I would say, is art of asking questions. And we should always keep asking questions. I think Indian students somehow are hesitant because of cultural reasons. They don't ask questions. I think that's one of the thoughts that I have. Then you have to have divergent thinking. Divergent thinking means to reach a goal, you need to think of what possible ways I can reach to a solution. And that probably will help us. There are convergent ways of thinking also. For example, we know 8 plus 11 is equal to 19, but so I know I have to reach 19. So I would have to think of how do I reach 19. So these are two ways the philosophers think of creating critical thinking. One is you can think of divergent ways or convergent ways. Convergent ways is when I know my goal. Divergent ways is I need to reach a solution and I can take any other path so that I am able to reach to the desired goal. Brainstorming is important. If I have an issue, we should discuss and give priorities, and we should leave all room for imagination while we're doing brainstorming. And I would say it has been seen that at times you get ideas from unexpected places also. The brainstorming is critical for, again, critical thinking or innovation, I would say. And there is no question, there is no idea that is too silly. You might have seen what was thought to be imagination now is a reality. We are also discussing things, meeting through this virtual seminar, webinar. This may not have been imaginable 20, 25, 30 years back, but now it is a reality. So, and there's nothing which can be silly, and we have to think beyond what is obvious. When we have an idea, good to take feedback from others, solutions from others. So by doing this, we will be able to find a better way out of the problem for which we have a problem on the hands. Look at this is an interesting slide which I found today morning. It says 29 ways to stay creative. It says a lot of things which you might find weird. Take breaks. It's been said when I'm writing a manuscript, I should not work on the manuscript. I should take a break, come back to the same manuscript, do that again. It will work out better. Have coffee, sing in the shower. Even while I'm exercising, meditation, these all would help me to be creative. And there could be many, it says collaborate, get feedback, what I told you, listen to new music. At times it might be try free writing, write something which is useful, read something which is out of your field. It says read a page of the dictionary. Maybe you'll find a new meaning. I would say if you read a book of biochemistry also, let's say today, and you read it after 2 years, you will find a new meaning in the same paragraph. So creativity, there are many ways. I said 29 ways to be creative, or this person said 29 ways. You will find 101 ways of being creative, or even more ways to be creative. Who can be creative? Anybody can be creative. So you can see a teacher in the class or a student, when this interaction happens, this is a place where creativity can come. I brought Mirza Ghalib here. He was also creative. Poetry is a creative way. In kitchen also you can be creative. While you're writing something, you're creative. Even a person who is, you might have seen Indians are more creative. When we say jugaad, it is all creativity. And a cycle repair guy will also be creative. He knows when he's doing the task over and over time, how can he improvise on the same? So further going on, how do we develop creativity? Let your ideas incubate. Don't try to push the agenda too quickly forward. So give it a time, a thought, and then take that forward. And resist yourself for immediate reward. And you have to be self-confident, positive, and feel the joy of creation when you want to be creative. Stages of creativity. I think I said this all. It's almost, you can say, I'm trying to sum up. Preparation, incubation, illumination. Illumination means filtering of some of the thoughts while after you incubate that, and then you verify some of the facts, whether it's doable, how do I reach the goals, and things like that. And never believe your memory. You will say, I keep things in my head. As all scientists, I would say you should all keep records of whatever you do in your labs, things like that. When you're even innovating, you should keep records, because you can't trust your memory. If you trust your memory, you know the picture on the right would happen. And don't wait till tomorrow. If you can do something today, try to do it today only. This is an article from Frontiers in Neurology. The article says creative expression of science through poetry and other media can enrich medical and science education. So I'm not saying, whatever I'm saying is not, that is not validated, it is validated. You have articles saying that you need to have certain kind of hobbies to be creative. And probably it eases your pressure on your brain, or de-stresses you, or you come rejuvenated. These are things that would stimulate creativity, I would say. Putting your heart is an important as brain. So your heart and brain both need to be creative. And this, you all are familiar. There's a dialogue from a movie of Shah Rukh Khan which says, कहते हैं अगर किसी चीज को दिल से चाहो तो पूरी कायनात उसे तुमसे मिलाने की कोशिश में लग जाती है. So if you are positive and you have your heart in, probably you will find a way out also of this. Talking about innovations during COVID pandemic, I would say worldwide, there have been calls for immediate innovations to address thousands of problems directly and indirectly resulting from pandemic. We were all in homes. A couple of months back, we saw life was at standstill. And that turned out to be a universal wake-up call for all such innovative practices, scientists, clinicians, hardware and software developers, product designers, economists, innovators. Everybody was together to find ways to manage this crisis. And if you look at world is looking at India in its efforts to develop vaccine also. US even is saying probably the vaccine is going to come from India. And even we had a government slogan saying Atam Nirbhar Bharat, because we knew we cannot be dependent on any country when we have to fight these kind of pandemics. And there have been efforts that we developed a lot of indigenous technologies. You might have experienced that we had nothing when the pandemic struck the country. And I will talk about these innovations in different sectors. And some of them you are familiar. And some, if you're not familiar, maybe the other speakers, my predecessors, might have talked to you. The first and foremost thing we were talking when this pandemic struck, that flattening the curve. Flattening the curve means if the numbers are going to escalate like this, if we can flatten this, we probably will be able to handle the pandemic in a better way. And India was able to at least push the peak for a couple of months, and that probably helped India to at least set up certain healthcare initiatives that probably would be useful in handling the crisis. One of the things we did was, lockdown. I think in India, we had to have the paramilitary forces, police to come in and ensure lockdown. This was something you can say innovative practice that did not happen in other countries. We utilize drones for monitoring areas which were hotspots. We even use drones for spraying sanitizers, and there are other countries also probably that did that. This was a new way of handling the pandemic. Manufacturing of disinfectants. We knew these disinfectants were sold out all over the world within few days. Our industry was not ready to handle the demand for these disinfectants. And if you follow the news also, there are still challenges with that the people have been coming out with spurious disinfectants. Some of them contain methanol. And there have been FIR registered in a state of Haryana 13 or 15 companies making disinfectants with methanol. So there are, when I say there is a challenge, that means there are increased opportunities also. And I've seen there are entrepreneurs, even brewery industry is started making sanitizers because to meet the increased demands. So you need more of sanitizers, you need more of soaps. And people are even innovating, how do we dispense these, how do we make them more effective? And some of these innovations you can see that happened from India is a wrist sanitizer. You wear it on wrist, press a button, it sprays the sanitizer. Then there is a wrist strap buzzer. We all know how many times our hand goes to the face. And there is an innovator who's developed this wrist band strap buzzer. Moment you go with your hand goes to the face, it will alert you, and it will even count the number of times you've taken your hand to the face. So these are kind of innovations that we needed during this time, and there have been some progress. And this is decontamination sanitizing tunnel. If you followed the news, you probably are aware, but there are issues with this tunnel because of spraying of chemicals through these tunnels, which probably are risk factors. But yes, this was a thought that was given that we could develop this. Advanced wash basin sanitizers developed, automatic sanitizers developed. These were all required to make it the sanitization or disinfection more effective. And you know that we did not have these kind of facilities in the country. And if I don't know, can you hear me?
Yes, yes, you are audible,
Yes,
sir.
yes.
I don't know what happened to my slides. They're frozen.
In COVID-19, so many things are frozen. So the same thing is happening even to the internet also.
Yeah, I don't know. It's just maybe. Can I reboot maybe?
Yeah, yeah, yeah.
Yeah, yeah, yeah.
Hello.
Yes.
I'm sorry that this happened, and I, I have no understanding why this would happen. My mouse just froze while we were discussing. Anyway, technology has its own issues,
Yes, yes, sir.
and I was talking about how things have, how our lives are changing, and what kind of opportunities are there. We are coming up with now ways we can disinfect food. So you might have experienced while we were buying groceries, we were so scared to buy groceries from vegetable vendors, and you knew initial incidents that there were a spread of virus because of getting them from vendors itself. So we are coming up with new sprays which could be disinfecting food formulations, and there are innovations in the country which are activated hydrogen peroxide based sprays that are not harmful if they are sprayed on the food and then ingested. And India did not have enough masks to handle the crisis, and Indian industry had to shift their gears to meet the requirements for mask. And we were advised not to wear N95 or any surgical mask for the reason we did not have enough mask in the country available then. Now we've escalated the production of, and there are innovations that came from India. People have been thinking that if we can recycle these masks by disinfecting these and then reusing them again and again. And these are other innovations that came up. For example, you need disinfecting equipments in the offices. Also, you had to handle papers. How would you handle papers if these are not, there is a risk of getting virus from these patients, and the UV chambers that were developed within the country which could take care of these disinfect the papers, documents, and other things. And you would know that probably some of even the currency disinfectant were required for the banks to be functional. And these are some of the innovations that came from Punjab University itself. So Punjab University developed these face masks using 3D printers, and then we also developed these disinfectant box. Along with that, we also developed on the campus automatic hand sanitizers, which would be much cheaper and been provided to public at much cheaper rates than what is available commercially. Besides that, this was also another field of innovation, immunity boosting products. Even Patanjali came up with certain immunity boosting formulations, and there's a ample scope of I would say innovation in this. Even if you just follow the market, amla, which was available for 40 rupees, went to 400 rupees a kg during this time of COVID crisis. So and it's been said people who have better immunity will not have serious complications from COVID. So there is enough market for these kind of products also, and there are newer innovations coming up in this. Zinc, vitamin C are kind of things that are recommended in COVID. We did not have enough healthcare infrastructure in the country, and you are familiar with these pictures that I've shared with you. You are developing isolation wards within the stranded coaches in the country. Some countries use stadiums for converting them into isolation wards, and this is an inside picture of this coach which was made as an isolation ward. Schools, colleges, hotels been used as isolation centers. This is a new normal that we saw, and still our campuses, 3 or 4 hostels are still being used as quarantine centers. So the challenge is not gone. We still have to think of ways we can handle this crisis. Ventilators costing 10 lakhs, 12 lakh rupees. Now the cost has come down to 15,000. But there's an interesting development that, as you must have seen, now the thought is that we don't need that many ventilators. So every, I would say, fortnight or a month, the whole paradigm about handling COVID is shifting. So now they say we increase the mortality when we put them on ventilators. So unless there is a person is critically ill, we don't need a ventilator also. But innovations were there in this. This is IIT Bombay developing this indigenous ventilator costing probably 10-15,000 rupees, which was comparable in technology with one which is imported. But yes, the challenge still was, how do we meet the requirement of the country when we have a country of 130 billion people? This is again another ventilator developed in the country during the time of COVID. Indian Navy also came up with innovation. They developed innovative low-cost PPE equipment. Covet Safe, a transporting vehicle for COVID patients. We know that we don't want the aerosol from the patient to be infecting other people. So this was a chamber that was developed where you could maintain gases and other vitals of the patient while it's being transported and keeping that in isolation. And we all know probably to handle COVID, we need test, trace, treat, 3 Ts. And we did not have enough capability to test in India and to report the results quickly. So it needed, so the challenge was we did not have enough BSL-2, 3 labs. We did not have enough PCR machines. We did not have any kit that would be used for this. We had to depend on international companies for supplying us with the kits. We started thinking of changing research labs in institutes to testing labs. And there are examples where a lot of research institutes in the country are now testing labs for different states. The capability was limited. The cost of this test was 4,600. Now we are thinking of reducing the cost to 3,300. But I would say the efforts are on to even reduce the cost further. And other challenge with testing was reliability. We know what happened to Chinese kits. They were all sent back immediately within first week of their arrival because they were all giving false positives. And this is a news picture I picked up from news. India returns faulty test kits from China. So Indian innovation in this has been, we've been able to develop cheaper PCR-based kits. We have been able to develop cheaper antibody detection or antigen detection kits. And these are all innovations that happened in a period of 3 to 6 months. So during the time of challenge, I would say Indian innovation has also been at the forefront. This is a saliva test. This is a new innovation, not from India, but probably where you can directly pour saliva and then know the test within 5 minutes or 10 minutes. Then this is a test that was developed by Abbott Laboratories, where you put the sample, it analyzes PCR-based reactions. But look at what it says. It can only be used for US only. That means the challenge is you have to develop your own kits. For the reason the strain of virus changes also, mutates also. If you don't develop your technologies, probably you will stay limited also in terms of detection of this virus. This is an innovation. At airports, you develop these COVID whisks, which would be used for testing of these COVID patients. Then another, I said test, trace, and treat. Tracing is the most important thing when it comes to COVID. And Indians developed this app called Arogya Setu, which will alert you if you are near an area where there is a COVID positive person. And look at this. Once this application is developed, it says you will have access to health care because there are 500 million smartphones that would have these applications. If there's somebody having a health issue later after the pandemic is over, this app would also be useful then also. So it gives you 24/7 access to healthcare without any major cost involved, just an app-based system, which could even be tweaked for other applications later on. Increased opportunities in pharma industries. Look at screen time is increased. Pharma industry is coming up with more and more innovative products which could be good for your eyes. So this is where there will be increased demand. There will be increased demand for cosmetics. People are coming up with cosmetics which don't need any contact. You don't want to go to a barber because there is a contact. So there is a requirement for things that could be easy, contact-free, so that we are able to use them for different and hygiene products also, which are contactless. If they can be developed, there will be enough market for that. In therapeutics, the world is fighting, coming up with molecules for therapeutics and potential drugs. I will only highlight a couple of them: hydroxychloroquine and remdesivir. And then remdesivir interferes with the RNA machinery, whereas hydroxychloroquine, we don't know precisely what are the targets of this. There are other drugs which interfere with translation, and there are more and more molecules that have been tested. And look at something interesting happen with hydroxychloroquine. They said hydroxychloroquine is toxic. It has a lot of side effects. Now we know this publications have been retracted. That means there was somebody who was pushing the agenda that hydroxychloroquine be retracted. We should not let India take the advantage of prescribing hydroxychloroquine to the patients. As so, there are interests that would be pushing a wrong science forward also. And Lancet has apologized that they did push data without too much authentication. Then there is a race for corona vaccine, and the different kinds of vaccine molecules being tried, and nucleic acid based, virus based, viral vector based, protein based vaccines. And look at the efforts now. Virus based vaccines being attempted, 5 or 6 molecules in pipeline. Viral vectors, almost 25 molecules in pipeline being tested. Nucleic acid based, 20 molecules being tested. Protein based, 35 plus molecules being tested. There are other approaches, microRNA based approaches being tested, and few others. Look at world efforts. North America leading in efforts to develop vaccines, China following, Europe, and India is even in the race, and we are putting all our best efforts to develop the vaccine. And there are 2 vaccine molecules that have been come from India itself, Covaxin, developed by Bharat Biotech, and we are probably having these vaccines available in the market very soon. And the other one, Covishield, probably 100 million doses of this vaccine is being mass produced to be used. And these are innovations that happen within India only. We talk about herd immunity. I'm going to skip this. If nothing works, we'll have to wait for years, or maybe at least minimum 1 year for herd immunity to develop, so that we don't have fear of this virus. So, and the fear goes sooner, I would say. It has impacted our education. We know we are also meeting in a virtual world, and we know schools are closed. And a few days back, I gave a quote to one of the papers that when we do teaching in a virtual world, we are engaging the students, but teaching cannot be as effective as it can be through regular classroom teaching. So it has impacted education also. New normal is being developed. We don't know how the examination will be conducted. The country is meeting this challenge, and hopefully we have to come out stronger. And this is the virtual classroom that we see now. And I'm also doing the same thing. I would have been standing and sharing this content with you, maybe seeing you interact with me. And but there are challenges with that. Anyway, we have to adjust to new normal, keep this learning process on, and keep the students engaged. And I said these are challenges. Practicals, how do we conduct practicals? How do we have interaction? How do we conduct exams? Our examination system is all subjective type, and government says limit screens time. How do we do it? And economics, there are economic considerations also. And India has been said that GDP would be almost zero, and we don't know when this economy would boom back, recover or boom back to normal. And there would be a long phase of recovery. This is what is expected. Nobody goes to malls. Cinema industry is all gone, and there are other things. International flights, hotel industry, all are impacted, and there would be severe fallout of this on economy. Mass migration did impact the economy. We all familiar with this picture also. And there would be half billion people that will be pushed to poverty because of this COVID crisis. And impact of environment, positive things to talk to. But we know that this virus came from zoonotic disease. I'll not talk about this. Maybe my predecessors did talk about this. But when we impact the environment, this is what we get from changing the environment. And these are positive things of COVID. You see peacocks, birds, and you could see flowering was much better. Air quality was much better because of COVID, because we didn't have enough people, enough vehicles on the roads. This is Delhi before pandemic. This is Delhi after pandemic. This is a real science picture where you can see nitric dioxide levels in a certain part of China before quarantine. After quarantine, you can see how the polluting gases have been gone down during the pandemic. Socio-psychological perspective, and we know there was a speaker who talked about psychological issues during COVID. I'm just going to touch upon this. this is what we were sometime back. This is what we are now. And touch is very, very important in human lives, I would say. Socializing is important. And it says positive touch promotes dopamine release, which is good in many, many ways. It promotes serotonin release, releases oxytocin, keeps us happy, decreases cortisol. That means decreases stress. But when these positive touch is missing, we have problems, depression, suicidal behavior, cognitive developmental delays, immune system is also weak. And these are some of the things that have been attributed to pandemic, depression, mental health issues, domestic violence. Nationalism is also one of the things that we've seen. And implications of this pandemic are far-reaching to economy, to governance, to healthcare, to education. But there are opportunities also that come along, and we need to take that forward. And all of us being in science, we should also do something useful. This is an interesting site. I would like all of you to visit this site. It says COVID-19 Innovation Hub. And this innovation hub, where you can see all innovations are registered in this site, and you can access different innovations in different fields using this site. And it says COVID-19 Innovation Hub.org is the website which you all can visit. And there's a quote I would like to say by Charles Darwin. It says, "It is not the strongest of the species that survives, not the most intelligent, but the one with most adaptable to change that survives." And another quote from Kiran Majumdar Shaw, she says, "Ultimately, the great lesson that COVID can teach humanity is that we are all together in this fight." And that's probably we all have to do. And thank you all for your attention. This is one of the pictures of our campus, and this is called Gandhi Bhavan. It's an iconic building on the campus. Thank you all. Thank you.
Thank you, sir. Now, forum is open for the question. Thank you, sir, for very wonderful talk. And really, the situation is very scary, but together we can fight and we can overcome with this situation. And there are lots of innovation are going in different fields during this COVID time. And the best home take message from your lecture is, "Khush raho, lekin kabhi santush mat raho." This is very good for researchers, sir.
Thank you. Thank you. Thank you.
मेरा भी कुछ कहना है इस पे सर आपको। खुशी तो कोविड-19 ने ही छीन ली है।
बिल्कुल ठीक है, बिल्कुल ठीक है बात है।
ठीक है, और असंतुष्टि अपने आप आ गई है।
बिल्कुल ठीक।
It was nice hearing you. I think for the first time I'm listening to your talk, and it was very well covered. I think almost every broad things that need to be learned, or it should be imbibed and kept in mind for future, that has all been revealed by you in this wonderful deliberation. And on personally from the Department of Biochemistry, many thanks to you.
Thank
Over
you.
to Pathak, ma'am.
Thank you.
Thank you, sir.
Thank you.
Thanks a lot.
Take care. Thank you for everybody for attention, and thank you for having me here. Thank you again. Thank you.
Thanks a lot, sir. Now, moving further, we have our today's last speaker and last speaker of this international webinar, Dr. Nikha Jamal. She is professor and Center for Scientific and Medical Studies, King Saud University, Riyadh, Saudi Arabia. She did her master's in biochemistry from University of Madras, Chennai, and PhD from Dr. Ramanoloya, Awadh University, Ayodhya. She is alumni of our department. She started her academic career as assistant professor at Department of Biochemistry, King Saud University, and promoted as associate professor. Now she is professor. Her area of interest is biochemistry of hydroxyproline and collagen, heavy metal toxicity, and nanobiochemistry. She has published numerous research publications in peer-reviewed journal and authored various review articles. She is a member of various scientific societies like British Society of Matrix. Life, she is also life member of Indian Society for Parasitology. She is also the member of Indian Society for Biological Chemists, SBC. And with this, now I very humbly request Professor Nikha to deliver her talk on COVID-19 effects on scientific research. Over to you, ma'am. Engineer Amit, yeah. Hello?
Yeah, can you hear me?
Yeah.
Okay.
You are audible.
Good morning, everybody. At the beginning, I would like to thank Dr. Farooq Jamal for having invited me and inviting me to this seminar and making me a part of this very nice webinar. And I'm happy to be with the RML University again because I was I was an alumni in the sense, though I never studied there, but I was among the first students in 1993 to be registered for PhD when the department was just started. So, it's nice to be there after all these years. And my topic for today's talk is about the effect of COVID-19 on scientific research. So for the past 2 days, we have been hearing a lot, and we have gained immense knowledge about COVID-19 in different aspects, right from scientific aspect to psychological aspect, social aspect. And so this is just a small contribution to that. What is the effect of COVID-19 on the scientific research? So let me start with my talk. Okay, can you see my screen? Can you see my screen? Hello?
Not yet. Not yet.
Okay. Just let me, I'm trying to share the screen. Just give me a few minutes.
Sure, ma'am. Take your time. No problem.
So is it visible now?
Not yet. Yeah.
Okay.
Now it's visible. Yeah, now it's visible.
Okay. So this is the topic of my talk, that is the effect of COVID-19 on scientific research. I'll just give a brief introduction, though it is a repetition because we have been talking about it, and we have heard, we have had even increased knowledge and increased scientific information of COVID virus disease. And this was first identified in December in Wuhan, China, and this has resulted in the ongoing pandemic. And first cases, they have been traced back to November 2019 in Hubei. And as of July 2020, more than 14.5 billion cases have been reported across 188 countries and territories, which has resulted in more than 600,000 deaths worldwide. More than 18.13 million people have recovered. Some of the symptoms, they are fever, cough, fatigue, and in severe cases, it progresses to acute respiratory distress syndrome, which is precipitated by the so-called cytokine storm, multi-organ failure, septic shock, blood clots. And the time from exposure to the onset of the symptoms, it varies, but typically it is 5 days. The spread of the virus, it is mainly spread through droplets, which are dispersed during coughing, sneezing, talking, and even people may become contaminated by touching contaminated surfaces like doorknobs, door handles, and then touching their faces. The diagnosis, it is by real-time reverse transcription polymerase chain reaction from the nasopharyngeal swab. And as we say, prevention is better than cure. The prevention measures include washing hands, maintaining physical distance, quarantine, the so-called social distancing, and the use of face masks. Right now, since there is no treatment for this disease, it is only management of the disease, and the management includes treatment of symptoms, supportive care, isolation, experimental measures. The World Health Organization had declared COVID-19 outbreak a public health emergency of international concern on 30th January 2020, and it was declared a pandemic on 11th March 2020. And local transmission of the disease has occurred in almost all the countries of the world. The national responses, how countries have responded to this pandemic, most of the countries, they have restricted cross-border movements, and the European countries, the Shenzhen area, they were the first to impose these border controls. This has been followed by quarantine and curfews, stay-at-home orders, and lockdowns. By late April 2020, we saw that almost 300 million people were under lockdown in different nations of Europe. The same number, almost 90% of the people were in some form of lockdown or other in United States, Philippines, and in India also about 1.3 billion people, they have been in lockdown. Coming very briefly to the impact of this disease, it has been a major destabilizing threat to the global economy. Millions of jobs have been lost. Tourism has been affected. There has been a storage shortage of essential supplies. Cultural and performing arts sectors have been affected. Politics has been affected in that in many countries and at different times we have seen there has been suspension of legislative activities. Education has been affected. Schools and colleges, universities have been closed in response to the pandemic. Then we have had other issues like there has been reduced visit to hospitals, shortage of health supplies, negative impact on the mental health, which we had seen in the yesterday's lecture, how the mental health has been affected. There has been an increase in prejudice, xenophobia, and racism around the world. And along with this, last but not the least, even the scientific research has been affected. Now, coming to the effect of COVID-19 on scientific research, we have seen, according to a recent study which was published in Nature Human Behavior, there has been a decline in the working hours of a scientist, with the average number of hours declining from 61 hours per week pre-pandemic to almost 54 hours during the pandemic. Now, it has affected different aspects of scientific work in a different way. Scientists, they perform different work like research, writing papers, planning experiments, fundraising, writing grant proposals, teaching, administrative duties, editorial, and clinical duties. So among all these different work which are done by the scientists, the part of the work which has been most affected is the time which has been devoted to the research. This has decreased. It has decreased by almost 11% on an average. And the work, the time devoted by the scientists to the work has decreased by 11%, and the greatest contribution has been the time devoted to the research, which has decreased by almost 24%. Scientists working in different fields, they have been affected differently. Not all have been affected by the same way. And the fields which rely on physical laboratories are the ones which are hard hit. And sciences like biochemistry, biological sciences, chemistry, chemical engineering, these are some of the fields which have reported the largest declines in the research time, with the range from 30 to 40%. On the other hand, the fields like mathematics, statistics, computer science, economics, they have reported lowest decline in the research time. And when we come to the gender of the scientists, we see that the female scientists, they have reported 5% larger decline in the research time as compared to the male counterparts. And among this also we see that the scientists who have at least one child of 5 years or younger, they have reported the larger decline of about 17% decline of their research time. And the second aspect is that this pandemic, it has caused a drastic decrease in the academic research, research in the industry, government aided research in basic and clinical sciences. And most of the resources, they have been directed to the research on COVID-19. Most of the clinical trials, they have either been paused or they are going at a slow pace. Some of the ongoing trials, they have been modified so as to enable the home administration of the treatment, virtual monitoring of the subjects to minimize the exposure of the subjects to COVID-19 infection. The long-term effect of COVID-19 on research are yet to be seen, but we have started to feel some of its effect on our work as a scientist. Some of the scientists may feel stuck because they are unable to carry out their experiment because of the COVID-19. And this is not only due to the fact that their workplaces, the universities, the labs are closed, but also due to the need to look after the children because the schools are closed, the kindergartens are closed. This is especially serious in the case of young scientists who are on short-term contracts and they have got a reason to worry about their future careers. According to a study which was published in Genome Biology, 57% of the scientists reported that they had lost some of their work, and they will have to repeat the work once the labs or the universities reopen, and this will consume additional funding. 25% of the scientists reported that they lost about 1 to 6 months of work because of the laboratory scientists. And this was mainly seen in the case of wet lab, where about 73% of the scientists were affected. And another factor contributing to the decline in the working hours of the scientist or the research contribution of the scientist was the personal living condition. That is, if they were living alone or they are living with a spouse or with a family. And the second factor was expatriates, especially the trainees in working in life sciences in the institutes which were located not on their home country but in foreign country. It was observed that the young scientists who were living alone, they had almost, who were living in a foreign country, they almost lost 40% of their research hours. In the case of expatriates, they may feel more isolated because of the language barrier and frequent updates from the local governments from time to time. Then it was also seen that the female scientists, they were reporting fewer productive hours. This was due to the fact that we have a higher number of female scientists working in the wet labs, and also due to the child care duties, which I have mentioned earlier. So this again demonstrates the vulnerability of the female scientists during institutional shutdowns. But on the other hand, this pandemic has intensified research challenges. There has been a rush and a competition among the scientists to publish their manuscripts, news reports, blog, and to such an extent that many cases, they don't even meet the clinical standards set by the FDA on COVID-19 specific standards. And in many cases, we have seen that the research have been published without peer reviews. Misinformation from the studies has resulted in diversion of resources from development and testing of more therapeutic candidates, and it has endangered lives. An example being ibuprofen. Initially, it was reported to be unsafe for the patients with COVID-19. So this resulted in a shortage of acetaminophen, and which was endangering individuals for whom it was needed. Similar was the case with hydroxychloroquine, which was initially reported to be effective for the treatment of COVID-19. So this resulted in shortage of hydroxychloroquine for the patients with autoimmune diseases to whom it is administered. Then a number of scientific conferences, they have been canceled or postponed. So this is just a snapshot of how COVID-19 has affected the life of a scientist. We see that 77% of the life science institutes have been shut down, 57% have lost about 6 months of work, and then about 26% of the scientists feared additional cost due to work loss. But on the positive side, we see that 66% of the scientists, they use this time to write new research proposals. 33% of the scientists, they attended virtual conferences, and about 80% of them took part in COVID-19 related researches. Now coming to the brighter side of the effect, we can see that there has been video conferencing, which has gained importance in running group meetings, journal clubs to meet the collaborators. Most of the trainees, they have taken this opportunity to learn new subjects like bioinformatics, programming languages, etc. Scientists, they have also been, they have begun using this time to develop new scientific projects while working from home. And a very frequently cited example here is that of Newton. So Isaac Newton, it is said that he had developed most of his theories on classical mechanics when he was working from home during the plague outbreak in 1665. Then coming to the patterns of collaboration during COVID-19 outbreak, we see that science today is internationally connected and it is being increasingly collaborative. There have been means to work from home on computer. We are able to connect with the colleagues locally, globally through video conferencing system, teleconferencing patterns, collaboration tools like Slack. For computational biologists and data scientists, collaboration is being facilitated through electronic means, and there has been collaboration in co-development of computational methods. And then we have seen a spurt in the new collaborations, online activities like journal club, virtual science seminars, workshops, training activities on topics of common interest. Most of the scientific conference, as our conference also right now, it is being held as virtual meetings. the 2020 edition of Biology of Genome, it was run online with real-time streaming of talks and moderated live discussions. This has also facilitated the engagement of researchers in collaborative activities directed against COVID-19. In the European COVID-19 data portal, there was an announcement by the president to help the scientists coordinate in sharing of the research data using European Open Science Cloud. There has been a rise of preprints. A preprint, it is a complete scientific manuscript that is uploaded by the authors to a public platform. The preprint, usually it is an earlier version of a manuscript which has been submitted to the journal for peer review and publication. After a brief quality control, this manuscript is posted on the preprint platform without a peer review, and it can be viewed by almost anyone in the world. So this allows the scientists to directly control the dissemination of their work to the scientific community worldwide. So we see a number of relevant preprints related to COVID-19 which are being posted on a number of preprint platforms. And here these are some of the preprint platforms related to COVID-19. One of the advantages of preprint over the traditional peer review is that there is a scrutiny from a large number of scientific population than provided by the traditional peer review processes. But then the danger comes when the report it is interpreted as definitive. Now we have seen a rapid growth in the preprints as scientists compete with each other to put their findings on the public domain as quickly as possible. In April, almost 17% of the COVID-19 publications, they were as preprints. And these preprints, the data from the preprints are, they are disseminated by the media also equally rapidly. So there is a risk in this case of generating and communicating the findings which are not robust. And we have also seen the result being that there has been a retraction of number of COVID-19 related research products. These are some of the platforms, virological.org, which are being used for the communication of the scientific data. Imperial College reports are being translated into multiple languages for the dissemination of the scientific knowledge. The global pandemic has highlighted the importance of research, of stable research infrastructure, funding for public health agency, disaster preparedness, response, and resiliency. So with this, I end my talk. And thank you all for listening to me. Thank you.
Yeah, thank you. Thank you, Dr. Nikha. Now, participant, do you have any query you can ask? Ma'am, very wonderful talk.
Thank you, ma'am.
And you rightly said that prevention is better than cure, and situations are really changing, and like there is a crunch of funding, there is a crunch of time, and like in India, total funding thing is going to be reduced as government has diverted all the money for the welfare. And but there are some silver lining, like we are talking to you via virtual mode, and we can increase our collaboration, and we can utilize our time for some some
collaborative.
We can start some collaboration, and we can start some, like for the life sciences people, this is very tough because our labs are not normally functioning. Then we can start some bioinformatics work like this, and it is
really, an opportunity to learn.
Yeah, it is an opportunity, and this is nice to meet you.
Thank you, Dr. Pathak. Thank you.
Thank you. Now over to Professor Jamal. You're not on. You're on mute. You're not audible.
Just a minute. Yes, yes.
Yeah,
I have
yeah.
unmuted myself.
Yeah.
Okay. Well, it was a nice deliberation from Professor Nikha Siddiqui. She is from Saudi Arabia, and she's a wonderful person, and she has done a lot of research also, and she is, I think, editor of several journals too. So it was wonderful listening to you. I think around 25 years or so have passed, and it's good that you got connected to this department after a very long time. Though personally, at personal level, you were connected with many people, with many faculty members of this department, but on a platform of intellectual talk, this I think was the first attempt we made, like to call you, invite you for your own presentation. So thank you very much. I think, do you want to say anything? Then I should.
I would like to thank you, Dr. Farooq, for integrating me back to the department of after almost 25 years. And it feels nice to be back with the department. Thank you.
Need not worry. Once you are integrated, you will remain integrated till we are there. Okay. Anyway, this 3 days international webinar has many interesting speakers, many distinguished personalities, and I hope it was enjoyed by one and all. So a very short time, all the participants who have joined the WhatsApp group, they will be receiving a feedback form, and do fill that feedback form, because that is the basis for your certificate. You will be getting a certificate in due course of time, and all the speakers, right from Dr. Prabodh Kumar Trivedi to Hemchandra to UN Dwivedi to Sunit K. Singh and Mohammad Haris, Amita Sarkar, Swati Vajpayee, Gunjan Tiwari, Rajat Sandhir, Nikha Jamal. Professor Ramashankar Dubey was very busy. Otherwise, we could have also heard his talk, listened to his talk, and that would have been wonderful. Actually, he is busy in some administrative work, and so in the morning he called me up and said that it was not possible this time. Better luck next time. Thank you. And then such sort of activities, international webinar, I think in true sense it got international because many of the speakers were from various parts of the globe. Okay, we tried to include as much as possible because the platform that was used was digital, and so on digital platform the enthusiasm is a little bit less as compared to physical way where we host and conduct international webinar or national webinar or something like that. So even I too was very busy because B.Ed entrance exam is to be held on Sunday. Despite all COVID-19 pandemic fanfare, still the state government is adamant on getting through with this B.Ed entrance exam. And I think this will again give a boost to the number of cases that we are witnessing every day. I personally take the opportunity to thank Professor Neelam Pathak, who has been, very well integrated herself with the time and very well not only cooperated is a small word. She has been a part and parcel of this international webinar. And my other colleagues, Dr. Sangram Singh, Dr. Neelam Yadav, Dr. Vandana Ranjan, Dr. Shivi Srivastava, they have been very helpful. They have been very cooperative. They have helped this international webinar to materialize well. And the number of the participants also was more than what a WhatsApp group provides you. It rose to around 256 or something like that. And then once again, this is not a official or a formal thanks. That formal thanks will be delivered by Professor Neelam Pathak. because I was the convener of this program, and we both were the conveners of this program. And so it was, I feel really privileged, and I feel really enthusiastic to say these few words of thanks to all the dignitaries and special personalities who have given talk, and all the audience, all the students, research scholars, media persons, and the technical team, and IIT, Institute of Engineering and Technology. My informal thanks and gratitude to all those who have been a part of this international webinar. Thank you very much. Over to Professor Neelamta.
Thank you, Professor Jamal. Now, I would like to request participant to provide some feedback. Participants, please.
Do we have a feedback form, or it is just oral?
Just oral. Please share something.
Okay, so you want me to start? Can I be?
Yeah, you're most welcome.
Okay, I mean, it was a very good conference. It was nicely organized, and the topics were very rightly chosen for the occasion. And it was indeed a very informative, scientific conference in every aspect. And we had gained a lot of knowledge, and plus meeting some of the old colleagues, and strengthening the forgotten colleagues, the bonds between the colleagues. So it was overall a very satisfying session, both scientifically and also socially. Thank you.
Thank you. Thank you. Thanks a lot, ma'am. Dwivedi sir, are you here? Sir? Anyone from the students? Otherwise, I am going to call the names. Hello. Hello. Yeah, you're
hello,
welcome.
Hello, ma'am, and this was very nice webinar. And I must say that is a complete package of scientific, psychological, and the innovations. Everything was there. So it was a very useful for faculty members as well as the students also. So thank you very much. And we hope that in future also we will get good from your institution, from your university, so that we can learn a lot. Thank you, ma'am.
Thank you. Thank you, Dr. Nia. Dr. Meenakshi, are you here? We want to listen something from you. Hello. Hello. Dr. Manikant.
Manikant sir, unmute yourself, please.
Hello. Good morning, ma'am.
Hello. Good morning.
Ma'am, overall, all the sessions are very informative, and I am very much thankful to all the presenters, and we learned a lot from all the lectures. Thank you, ma'am.
Thank you. Anyone else want to say something? Or otherwise,
Ma'am, Shamshad sir want to say something.
Yeah, you're most welcome, Shamsad sir.
Hello, can you hear me, Dr. Neelam?
Yeah, yeah. You're most welcome.
So,
Yeah.
I'm Dr. Shamshad. I am working here in Sultanate of Oman in the Ministry of Higher Education as a
Nice.
assistant. I have been listening to all the talks, and I actually don't call me sir because I am your student. I have been a student of Dr. Ram Lohiya
Oh,
University as a
thank you, Shamshad.
master's. Yes,
Okay.
I have done masters in biotechnology institute, that
Nice.
university. So all the talks were very engaging. I really enjoyed the talks, especially by Dr. Singh Saab from BHU and Dr. Haris Saab. I really enjoyed those talks. Very interactive sessions out there, and these kind of webinars should be arranged months or yearly, so that all the alumni of our university can meet on the same platform and share their knowledge and help the other students, other budding researchers and students which are preparing for their future research endeavors or for the jobs. I think it was a very, very nicely arranged webinar. I must congratulate you and Dr. Farooq Jamal for taking the efforts. Thank you so much.
Thanks, Shamshad. I think Dr. Tausif is here. Dr. Tausif, Dr. Nawaz, Dr. Tausif.
Yes.
Yeah.
Quite
We want to
interesting.
hear something from you.
No, it was very knowledgeable. Unmute आवाज आई?
हाँ, yeah.
हाँ, तो अच्छा इंफॉर्मेशन थी जो गैदर की गई थी इस परिप्रेक्ष्य में जो आजकल चल रहा है, मगर ये है कि ये थोड़ी सी बायोकेमिस्ट्री वाली चीजें और होनी चाहिए थी. That I want to say. It is not a subject specific.
Yeah, actually it is a complete package for COVID-19. Most focus on
that is very good. That is very good. I just appreciate.
ऐसा मुझे भी कुछ कहना है. ये सेमिनार देखिए ऐसा है कि वो 3 दिन का था और 3 दिन में वो बायोकेमिस्ट्री भी आई, टेक्नोलॉजी भी आई, ठीक है, और तो वैक्सीन तक पहुंच गए हम लोग. ठीक है.
It was very interesting. Not at all.
मतलब आपका सजेशन सही है. हमने पहले ये सोचा था कि हम इसको आई शुड मेक इट मोर स्पेसिफिक, बट स्पेसिफिक में ये होता है कि नंबर ऑफ द पार्टिसिपेंट्स दे डिक्रीज एंड
yes, exactly, that is true.
ओरिएंटेशन, बायोलॉजिकल ओरिएंटेशन दे स्टॉक. तो इसमें साइकोलॉजिस्ट एंड साइकोथेरेपिस्ट, काउंसलर्स फ्रॉम अक्रॉस द ग्लोब, popular lecture type, and everything was just like Professor Nikha said, it was a complete package for COVID-19.
That is very true. Overall, it was very interesting, and all of you have the creditability for this to arrange such a nice meeting, webinar. And Neelam Pathak especially needs sincere congratulations from me because she has encouraged me to attend this webinar.
Thank you. Thank you.
Okay.
Thank you.
All the best.
Okay, thanks a lot. Okay,
yes.
Thank you.
Okay.
डॉक्टर नवाज, आपसे भी कुछ सुनना चाहेंगे हम लोग. Dr. Tuhina is here. Dr. Tuhina? Okay. Anyone from student? Last.
Vijay Shukla, I get you. Unmute yourself. Ask to unmute.
Mala, she has joined from the Chicago. Mala, do you want to speak something?
I think most of them are in the mute state. That is
yes.
Yeah.
Engineer,
that's the problem.
Vijay Shukla ji, आप बोलिए.
Yes sir. Thank you very much sir for organizing the conference, especially Farooq sir and Neelam Pathak ma'am. We got to learn a lot from this type of lecture. Thank you very much sir.
Okay, keep it up. Okay. Thank you. So I would request Professor Neelam Pathak to conclude. finally, with few words of
Thank you all. Okay. Thank you all. Thank you all. Our thanks is due to all the distinguished speaker. And thank you each and everyone from the organizing committee, especially the faculty member of Department of Biochemistry. And we are very much grateful and thankful to our technical team, including Engineer Ramesh, Engineer Paritosh, and Engineer Shobit. And finally, we are at the end of the webinar, and just I want to share a few lines of Robert Frost. The woods are lovely, dark and deep, but I have promises to keep and miles to go before I sleep, and miles to go before I sleep. Thank you all. And if Engineer Ramesh is ready, then we can just end this webinar by playing national anthem.
Just a minute, one more thing I would like to add. The feedback form is getting ready, and very soon it will be available on the WhatsApp group. So don't leave the group, because that group will be the basis for your certificate also. Okay, and we will get inputs, your real inputs from that Google form. Okay, so don't leave it till you fill up the Google form. Now we can, I request Engineer Ramesh Mishra to play the national anthem, and with that we will conclude.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Now I request Engineer Ramesh Mishra. Ramesh Mishra ji, can you hear me? सुनाई दे रहा हूँ मैं?
जी सर, बिल्कुल सुनाई दे रहे हैं सर आप.
These 3 days brainstorming session and international webinar on looking at life differently, fallout of COVID-19 pandemic comes to an end. So we hope that in near future, again, we will meet very soon with some wonderful personalities from across the globe, and such deliberations will find your way, and you will really be benefited both at the scientific level and also on the otherwise also who are from the non-scientific background. They will also be equally benefited, because we will try to plan the things in such a way that it will be an integration and amalgamation of many different types of speakers from different areas taking up different issues. So once again, the final thanks from the bottom of my heart to all the dignitaries who spoke, and all the participants, and all the members of the organizing committee, advisory committee, as well as the technical committee. Thank you very much. The session comes to an end, and Ramesh Mishra, please
Okay, sir.
do the needful.
Thank you.